Alright students, I'm going to talk real quick about section 3 and some of the points that were in there. I'm going to start with Mr. Sutton and his research on grasshoppers, which was pretty groundbreaking stuff that led us to determine, or let him determine, that the chromosome is definitely what is carrying traits from parent to offspring. His research was on the number of chromosomes in the sex cells versus the body cells of the grasshopper. Counting the chromosomes, he found 12 pair, or 24, in a grasshopper body cell, but only 12 singles in any grasshopper sex cell. So if you do the math, you see that a 12 chromosome sex cell plus another 12 chromosome sex cell, when they make the new grasshopper, makes the normal body cell count for 24 chromosomes. So half come from the mother, half come from the father, some from the sperm, some from the egg. So they always, of course, appear in pairs when they're in body cells, and always appear singly when they are in a sperm or an egg. So if mom and dad have, let's say, a big A and a little a allele, the big A may go to one sperm, the little a will go to the other. So they always split during the process of meiosis. Now, genes are carried on chromosomes, and that is a pretty good theory. It's called the chromosome theory of inheritance. And it basically states that all traits, all genes, are carried from parent to offspring on the chromosomes. Genes are carried from parent to offspring on chromosomes. Now, meiosis is the process by which we make sex cells by cutting the chromosome number in half. It's a pretty straightforward process. Some of it's a little bit tricky, the way that the numbers change, but you start with a single cell with the normal 2 chromosomes per. One chromosome always appears in pairs, so you have 2 for the body cell count. But before we enter meiosis, we double that, so we end up with 4 copies of every chromosome. Then, during meiosis, we're going to split them, 2 to one cell, 2 to the other cell. So then we have 2 cells with the normal count. Then they split again. Then we get a sperm or egg with 1 chromosome each, another sperm or egg with 1 chromosome each, another, and another. So every run of meiosis gives you 4 cells, sperm or egg, with only 1 chromosome copy each. So for humans, that would be 23 chromosomes instead of our normal 46 or 23 pairs in our sex cells. So a Punnett square, recall a Punnett square, as shown here, shows us meiosis when it shows how the parent alleles split this way and that way, and it shows us when the female parent alleles split this way and that way. So the side and top show meiosis, and then fertilization results appear in each of these little squares. So we show meiosis here and here, fertilization results inside the boxes. And that's why a Punnett square is so useful to us. Yeah, so that's what happens during meiosis and why we use it. Now, the way the genes are lined up on a chromosome is important. For every organism in a species, the genes are in the same order. Okay? In dogs, they have to be in the same order. In cats, for all cats, they have to be in the same order. All humans have theirs in the same order. Very important. Now, the 2 chromosomes in a pair will have the same genes, but they can have different alleles on each one. For instance, I may have my dark hair allele on one, but then I may have the blonde hair allele on the other. But since my brown hair is dominant, the blonde doesn't show up. Now, they say chromosomes are joined together like beads on a string, and the meaning of that phrase is that they're complete. We have 1 full chromosome, or 1 full gene on the chromosome for hair. We don't split it in half and have half a gene for hair, then the gene for fingernails, then the other half gene for hair. It's always complete. 1 full gene for hair, 1 full gene for fingernail, 1 full gene for eye, or whatever is next. And they come together one after the other like beads on a string. Of course, they say in the book about 35,000 genes in a human. I think that number has gone up quite a bit since this book was published. And of course, we can always expect that to be changing. And of our 2 chromosomes, like in the book, some can be homozygous, and you can get big A, big A. But like in this, we got heterozygous for A, because we have big or little. Heterozygous for B, little b, big B. But then we're homozygous for C, big C, big C. So we can have different homozygous or heterozygous for all the alleles on a chromosome, because the 2 different chromosomes, while they have the same order, can have different alleles for each of these genes. So that's a real quick look at section 3. that focuses on meiosis and the chromosome counts of grasshoppers, which is what showed us that chromosomes carry the genes. In a few seconds, we'll shoot a video for section 4, which is going to wrap up the molecular end, where we'll look at mutations and how proteins are actually made from the DNA.